Thank you very much, everyone, for joining us for our short little webinar sponsored by Kura Biotech and DPX Technologies. Um, this guy, waving, is Jack Andrews. I'm the business development leader for Kura Biotech, and the other guy on screen is Mr. Nick Chostara. So he's a application specialist for DPX Technologies, and we're very excited today to bring you an innovative approach to testing your urine drug testing samples. So I promise no sales pitch, only just a couple sentences here about Kura Biotech and DPX. So Kura Biotech, as the name says, we are a biotech company. Our core expertise lies in the design, development, and production of cutting-edge enzymes. The company as a whole wants to provide enzymes to laboratories that far exceed what you're used to getting, and we'd like to break the classic molds of how people use enzymes in their production environments. Today, we're going to hear about B1, and that's a great example of a nice piece of engineering to change how we think about using beta-glucuronidase. DPX, these guys might just be the best kept secret in solid phase extraction. So they are the world leaders in cleaning up any kind of sample that you might bring to the laboratory, actually within a pipette tip. So their patented in-tip dispersive solid phase extraction materials use classic chemistries as well as esoteric or customized designs. So their tip-on-tip filtration products is very much set to disrupt the market and how people look at filtering their samples. So just two ground rules to cover with you. So one, we are recording, and we have a chat box in the side there for you. So if you're having a technical issue or if you have questions, please let us know there. We're going to go through the entire webinar, and then at the end, we'll pick up any questions and answer those as time permits. Once that's all covered, we'll condense all of the questions, all of the answers into an email and send that back to you guys with a link so that you can replay the webinar if you missed something. And please feel free to forward this to any of your friends and colleagues that you feel like might benefit from this type of technology. So, all right, all the stuff aside, we can move on to what I consider the fun stuff. So it gives me great pleasure to introduce Nick to this audience. So I'm pretty sure that 15 years ago when he started his career in a GLP laboratory, he didn't realize that he was going to have to be the soup to nuts guy. He was telling me about actually receiving samples and resolving discrepancies all the way through looking at sample prep and method development, creating reports and sending them out. And that gave him a great foundation for the next phase of his career, where he spent 8 years at a Millennium. So there he was able to hone his sample prep and LCMS detection analytical prowess. And all of that combined really puts him in a great position to be an application specialist at DPX. I know he's been traveling around the West Coast for the last year or so, helping labs set up workflows and get their technology set up to be the most productive possible solutions in those laboratories. So without further ado, I'm going to hand the mic over to Mr. Nick, and you guys have fun.
Thank you, Jack. Thank you for the introduction, and thank you for everyone joining us today for this webinar. Today's webinar is going to be on load and go urine drug testing. Load and go. We want to load our samples and just let it go. So what I'm talking about here is a completely hands-free workflow, complete with enzymatic hydrolysis, complete with sample preparation, and a final product that is ready for injection. As Jack mentioned, having worked in a high-throughput clinical lab, I was developing methods, and we're always trying to find ways to speed up the process. We're developing more than just a test. We're actually creating a process. And I've always been an advocate for the sooner you get the samples on the instrument and get the instrument running, the better it is. The better it is for everyone involved in this process. So how can we do this? How can we speed up that process without sacrificing any of the data quality? How can we, in fact, load and go? So to get started here, some of our discussion topics, I'd like to take you through the load and go process. First, we're going to discuss the challenges of urine drug testing. And then I'll briefly go over a current generic workflow model and its limitations. Then I will introduce 2 new technologies to demonstrate how we can overcome these offline limitations. Then I'll go over to the design of the study, this proof of concept for load and go, and we'll take a look at the results. And time permitting, we'll have a Q&A session afterwards. All right, so jumping right in here. What are the challenges for the clinical laboratory? Well, on the corporate level, as everyone knows, money is everything. Money, money, money. It's not shocking to hear this. It always comes down to money. Reimbursements going down. Billable doesn't always equal payable. And we're always looking for ways to lower the cost of running production. Now, for a production standpoint, we have turnaround time. We have turnaround time, and we are increasing throughput, yet we are doing so with less people. But how do we maintain the data accuracy and quality? Well, time is money for production. So let's look at this workflow here. So this is my workflow model. Starting off, sample accessioning. So FedEx brings in the cups, UPS brings in the cups. We are transferring our urine samples into tubes. We're barcoding. We're logging in on all the information. This is a very difficult process to automate. It's very time intensive. And again, we are trying to get the samples on the LCMS as fast as possible. So our current workflow on the LCMS gives us comprehensive panels. So we're doing 80 compounds for 80 samples, and we're giving all this data to the data analysis team. And again, this is a very difficult process to automate. But let's take a look at the sample processing stage, the hydrolysis, pre-treatment. Most of this is taken offline from a liquid handler, but maybe we can take a closer look at that and see if we can't improve the workflow. So here I have a hands-on approach for the sample processing. We get our sample from accessioning, we run it over to the liquid handler. Liquid handler does its pipette command, and then we bring it over to the incubator oven, and we put our samples in, we set our timer, and we go to lunch. All right, well, everyone knows enzymatic hydrolysis is the gold standard. We are trying to make this assay less complex. We want to make it more sensitive, and we want to make it more cost effective. But this hydrolysis offline is still a slow bottleneck for the lab. All right, moving on to sample processing, another hands-on process. So after lunch and the hydrolysis is complete, are we doing any further dilution? Are we doing any SPE or filtration to the samples? We need assay robustness. We want less instrument maintenance. We want longer column lifetimes, and we want easy to review chromatography. So we must have cleaner samples to do this. Again, this process is very hands-on and is taken offline. So how do we take this hands-on approach and make it hands-free? Well, the answer to that is let's take advantage of the liquid handler. Let's bring some of these offline processes and let's put them back online and utilize this investment. So I had the excuse me, the honor of meeting Jack Andrews at MSACL back in 2019. I was presenting to him my poster for DPX on tip technology and showing him how we can automate sample cleanup. So he in turn introduced me to the B1 hydrolysis solution. And he asked me what I thought about hydrolyzing the samples at room temperature on the deck of the robot. I told him I had to see it to believe it. And sure enough, we came up with the load and go strategy. And now we can look at how to bridge this automation gap. So first off, what is B1? B1 is what I refer to as the solution. It is an all-in-one hydrolysis solution. This product is an enhanced, purified recombinant beta-glucuronidase. It's been engineered for speed and thermostability. One of my favorite attributes of this product is that the B1 actually comes stabilized in its reaction buffer. In fact, this product is actually a reagent, not just an enzyme. So it already comes mixed with the buffer. That's a real game changer. This product has tremendous stability. It has 3-month stability at room temperature. I mean, I can't tell you how many times working in the lab doing R&D where I've left the enzyme out overnight on my lab bench. I've come in the next day and I've had to basically just throw it away. I also see a lot of this master mix being produced where we're combining the internal standard and our enzyme buffer solution. And then I see a lot of this waste in production environments, just leftovers being thrown away due to stability purposes. So with this B1 product, we actually don't need to worry about any waste. We're not generating any waste. We can use every last drop of this solution. All right, tip-on-tip technology. What exactly is that? Tip-on-tip technology is a sample filtration technique using pipette tips to filter particulates from the matrices. And this is all done without offline centrifugation. The tip-on-tip product that I used in this study is a submicron low porosity filtration tip. This process does not require any positive pressure or vacuum manifolds. This is an automated alternative to centrifugation. It is ideal for high throughput labs. It's a low cost, versatile product that generates cleaner samples. And I have to say, this is quite a cool thing to see in action. All right, so let's review real quick. Using these two technologies, B1 and tip-on-tip, we can overcome some of these hurdles of the manual process, and we can start to create our load and go workflow. So B1 comes crack and pour ready. You add it directly to the samples. The B1 reaction is at room temperature, so now we can hydrolyze our samples in a matter of minutes on the deck of our liquid handler. And that would be the same liquid handler that we just used to aliquot the samples as well. Using the DPX filtration tips, we can avoid offline centrifugation step. We can increase the throughput and the robustness without FTE intervention. Now, depending on your robot, you can process 96 samples or less, and this process is done in less than 2 minutes. And again, we're not going to be generating any waste with the B1 solution due to its superior stability. So again, I just showed this slide, but the load and go strategy is we're using our liquid handler, we're incubating with B1 on the deck, we're using tip-on-tip technology to get cleaner samples. So let's jump right into what the study design is. What are we here for? All right, so we want to test the B1 hydrolysis. Okay, so to do that, I thought, well, if I can get codeine 6 glucuronide to hydrolyze, then I know all the rest of the panel analytes will be fine. Codeine 6 glucuronide is well known as the ultimate analyte in UDT. It's the most challenging to cleave, so I figured this would be the ideal target analyte for this study. I made 3 different sample concentrations that I spiked into negative urine, 500, 5,000, and for some sample labs, 25,000 nanograms per mil. Now this is a time point study doing incubation at room temperature for 5 minutes, 15 minutes, 30 minutes, and 45 minutes. And this is all done under competitive conditions. All right, so what do I mean by competitive conditions? I'm talking about competitive hydrolysis. Now this is something I feel very passionate about. This is the best way to test your conversion of your glucuronides. So for each of the codeine glucuronide spike samples I prepared, I included 100,000 nanograms per mil of acetaminophen glucuronide. This really challenges the enzyme in a realistic environment. So we have copious amounts of substrate, we have a target analyte, and then we're having B1 hydrolyze everything in the sample, and that's how we can stress and really perform a good study here. Some of the key components of the sample preparation technique. Yes, we are filtering the samples with tip-on-tip, as I mentioned. It's a very fast technique, but also I am using the liquid handler and this process to quench the reaction at each of the different time points, which is very important. So for my hydrolysis protocol, this is how I prepare the plates. I'm using replicates of each of my study samples, and I have 5 calibrators, 3 QCs, and I am doing 50 microliter aliquots of the cal QC and study samples for 100 microliters of our B1 solution. Again, the sample time points F5, 15, 30, and 45 minutes at room temperature. So let's talk about after hydrolysis. So the tip-on-tip aspect. So each reaction is crashed with methanol. So we're quenching the reaction using methanol. The liquid handler will add methanol to each sample cohort. It will mix by aspirating and dispensing. It will take a final aspiration of the entire sample. It will go over and it will grab our DPX low porosity filtration tips, which actually attach to the bottom of the tip that carried the sample. We bring the pipetting head over to our clean collection plate where it dispenses from one tip through the filter of the second tip and then into the collection plate. Those tips are then thrown away. New tips are picked up by the pipetting head. 100 microliters of this filtrate is then aspirated and transferred to another collection plate, which contains 400 microliters of water and is now injection ready. So just a quick overview. The method I'm using, the LCMF method, is in fact a validated comprehensive method that I use for anything from opiates, opioids, barbs, benzos, and even carboxy THC is in this panel. It's a 6-minute runtime. One of the key characteristics of my method I would like to point out, the upper limit of linearity is 20,000 nanograms per mil. This is significant for the study because when we are looking at the 25,000 nanograms per mil of codeine glucuronide, after cleavage, we're actually closer to 15,000 nanograms per mil of the codeine-free compound. So I know that my upper limit values, I'm linear through that concentration, so I know that the data is accurate. All right, so we have made it to the results section. let's take a look at this table I have made to check B1's performance. Now with the 500 nanogram per mil sample, we see at the 5-minute time point, I'm almost at 95% conversion, very low CV. This is very exciting. But if I move over to the 5,000 nanogram per mil and the 25,000 nanogram per mil sample, I'm seeing lower conversion and then a little bit higher on the CVs. So basically this is telling me that the reaction is still ongoing and that the 5-minute time point for the higher concentrations is not long enough. Because once we move down to the 15-minute time point, we see, basically full conversion of the 500 nanogram per mil sample. I am now at more than acceptable numbers for the 5,000 nanogram per mil sample at over 95% conversion. And I'm getting pretty close with the 25,000 nanogram per mil sample, almost 80%. And you can see here, it doesn't really improve much after that 15-minute time point. All right, so I also have this same data shown in a different table. This is a different graph, I'm sorry. This is a line graph, which basically shows me what I was just explaining, that after 15 minutes, we've pretty much exhausted under these conditions the hydrolysis. So what does this tell me? So labs that are seeking higher concentrations, such as SAMHSA labs, and they are requiring target values of, say, 15,000 nanograms per mil, I would certainly like to design a second experiment or study that can focus on the higher concentrations. Coming from a clinical lab, we really focus on the lower concentrations to make sure that we are not reporting false positives or false negatives. These hydrolysis reactions, as you know, are quite dynamic. There could be several things going on here that gives us these results. What we do know is that we can tweak this reaction. We can actually drive the conversion of the higher samples to full conversion. We have enough data that supports this, and just by tweaking some of these conditions, we are able to customize this hydrolysis procedure based on the requirements of the lab. All right, let's get to our conclusions then. So what do we have? We have an automated process with 15-minute room temp hydrolysis. This is possible. The data suggests that this is very possible. We also have GPX filtration, which can replace centrifugation and keep this online automated process going for cleaner samples. In my opinion, we certainly have acceptable recoveries of codeine, and our load and go workflow proof of concept has been completed. Now we do have a couple labs currently validating this process, and that we are actually customizing the hydrolysis protocol per lab. So that being said, this concludes our webinar today, and we have maybe one minute for a question. Jack?
So I think that first, apologies to everyone for the rough start. It wouldn't be a first-time webinar for us if we didn't have a rough start. So I appreciate everybody that hung in there with us and getting to this point. So we have the line for another 5 minutes or so. So if you'd like to hang around, I think we've got 3 or 4 questions here. Maybe we'll answer 1 or 2 of those. I understand that folks allot a certain amount of time for this. So if you need to go, again, we'll ship out the all of the questions and answers to everybody that's registered today in a follow-up email. So I'm looking through a couple of the questions. I'm just going to pick one here. So Nick, I see that somebody is asking about acetaminophen and the fact that that concentration is outrageous, and they want to know if you've seen acetaminophen concentrations in patients that high in your work.
Oh, it's a good question, Jack. Thank you. So in my history, I've actually never had acetaminophen as a target analyte. I'm only using it to add copious amounts of substrate to really challenge the hydrolysis efficiency of the enzyme. So in this case, I'm not sure if that's a clinically relevant number per se, but I know that acetaminophen is a very commonly used medication in our population, our testing pool, so that this is very commonly found in our samples. I might be on the high end of what those numbers are, but this is really just to challenge the enzyme efficiency, and I think it's done a great job at doing so. I hope that answers it.
Sounds good to me. So let's see. I have another question here. Let's do one more, Nick. This user is asking about how long does it actually take in those. So you mentioned putting this into production laboratories. His question is, how long does it take in a real production laboratory?
Yes, excellent. Great question. So this is a very customizable procedure. So if you have an 8-channel pipetting head, a 96 pipetting head, a 12-channel pipetting head, we can always customize the process around your equipment. But really, with the way it's worked out for one of our clients that has an 8-channel head, by the time it's done loading all the samples to the last row of the well plate, it's been 15 minutes. So the first row of samples is completed with hydrolysis, and it can start doing the filtration step. And then so on and so forth. So as we get to the 12th row, we are already at 15 minutes for that sample row. So with the hydrolysis and the filtration step, it doesn't really seem to matter if you have a 96 head or if you have independent channels, because we are basically waiting for 15 minutes with a 96 head, then doing the filtration step. Or we can be filtering all the other wells during that 15 minutes, and everything is actually pretty much done at the same time. So up front, depending on how long it takes to aliquot your samples, we're looking at about 30 to 35 minutes total time with hydrolysis, filtration, and actually setting up your plate prep with your samples, CALs, QCs, and internal standard.
Awesome. Perfect. Well, I see that we're approaching this 35-minute mark, and I just wanted to say again, thank you to everybody. There's like 3 or 4 more questions that are in the chat room there. We'll make sure to get those answered and sent out to everyone. But thank you again, everybody, for your attendance.
Yes, thank you very much.
Oh, perfect. Now's the perfect time to say, if you have questions, you can either reply to this email that's coming, or Nick and I have our contact information here, and we talk to each other pretty regularly. So if you have a question that comes back to the email, I'll make sure it gets routed directly. Awesome. Thanks, guys.
Excellent. Thank you very much.
Cheers.